thanking you for your participation and your engagement with our center, the Shiley-Marcos Alzheimer's Disease Research Center, which has been around now for almost 40 years. Really one of the very first Alzheimer's disease research centers in the nation. And I hope you all recognize from prior presentations how esteemed this center is. It really is an honor to be with this team of faculty and leaders in the country's Alzheimer's disease fight. And you're going to learn a lot about some of the new developments that are happening, some right hot off the presses, so Howard's going to be able to cover a lot of those burning questions that you're probably seeing in the news. I will save that for Howard to talk about, but really, really exciting advances. And it's been such a long time coming, really, to see some impacts on the progression of the disease. And that was really thanks to your participation, really. And it's the only way we can move this field forward is to have the engagement of our volunteers and our community. And it's so wonderful to see you and see your faces and see familiar faces and catch up. So I'm just going to give a brief update. I don't want to take any time from our most fun part, which is the interaction and question and answers session. But as you recall, our team is much larger than what you see here. We're all working together to kind of process the data, process information, the biofluids that you're so graciously giving to us to understand what the markers are for this disease and other diseases that are related. Whether you're a healthy control volunteer or a patient that's suffering from some of the cognitive impairment, all of it is very, very important to us. And so this whole team is gathered to really welcome you into the center and try to help keep you engaged and answer your questions. So this is our appreciation, and I should take this time to really send appreciation to Christina Gigliotti and Emily Littles who are really organizing this, and the rest of the staff. I always stumble on Gigliotti. I gotta work on my, is it Italian? I don't know. So work on my Italian. Okay, so this is our staff and other team members, but really also there's a whole set of faculty that are also that you don't tend to see a lot of, and that's because our center is organized in cores that really process all of the data, including the very, very critical donation at the end of life, the brain donation that so many of you have signed up for, again, so graciously, a critical piece to our efforts. So in the administrative core, myself, David Salmon, and of course Emily Little working with us closely. We have a steering committee with Doug Galasco, Diane Jacobs, Eddie Koo. We have a clinical core that really is probably the front end of the efforts that take place. That's where we're seeing you each year and processing the information that you give us and the testing that we do. And I know you spend a lot of time in giving that information to us. Be aware that we also spend a very long time trying to really make good sense of it as much as possible. Go ahead, would you like me to speak up?
Yes.
Yes, that's usually the request, so here we go. We have a data core that then will actually take all of that information, including the biofluid data, and make sure that other researchers across the nation, not just our UC San Diego scientists are using this information, but across the nation, the world's Alzheimer's and other related dementia research is making use of the information that's taken here, right here in San Diego. The neuropathology core I alluded to with the autopsy information. We have the biomarker core I introduced last time this is kind of a newer core because there's so much relevance to biomarkers, biofluids, cerebrospinal fluid, and really plasma now, which you may hear a little bit more about as well. There's a lot in the news about the excitement of now being able to get more information from the blood tests that we assess from you. Now we still need the cerebrospinal fluid, and I'm going to show you a couple slides of why that's so critical. But it's really exciting to see the advances in being able to take a blood sample and be able to understand a lot of what might be happening in the brain just from the blood. Again, the science that you guys have facilitated has really been pushing that forward, because Doug Galasco is a world leader in that, and he's right here with our team in San Diego. Human cell model core. This is quite interesting. In fact, you may have been approached about getting a little skin biopsy off the forearm. It's an incredible new model for assessing the genetic and the aging factors of Alzheimer's disease, to where we can now turn that skin cell in a dish into a human neuron. That's your neuron. You born it, right? You took it from your skin and we've turned it right into a neuron cell that we can now look at what are the impacts of genetic factors and aging factors in a dish on brain tissue. So not only neurons, we can turn them into other cells of the brain, things like astrocytes and microglia that all work together in this disease process. And it's really, I think, going to push this field forward, because up to now we've been trying to model this disease using rodents like mice, right? And it's often been stated, well, we've cured Mouseheimer's many times, but we've not cured Alzheimer's. So, and it is a very human disease. So this human cell model core that is now dedicated to this new exciting science of turning skin cells into nerve cells and other brain tissue cells, and even making what we call organoids. It looks like it's got all the factors of a brain in a dish, and you can look at what are the genetic factors that are impacting those really complex molecular pathways that are so critical to this disease. So really excited about the recruitment. I'll talk about that with Jerome Mertens, a new recruit that we brought over. He was trained at the Salk Institute, went over to Austria for a while, and we were able to bring him back. And we have a close relationship with the Salk Institute in that human cell model core. The outreach, recruitment, and engagement core, again, a place where we interact as much as possible with you. That's the group that is so critical to these educational events and other publications that we send out to you, the support groups, the Memories at the Museum, the Via Music, all those really hopefully fun events that engage you in our center and give you something to really connect you even beyond just the yearly visits where we collect the data. The Latino core, very, very important to diversify our cohort. We understand more about this disease in Caucasians than we do in the other diverse groups. And so it's very important to change that. Very important to be able to make this research relevant to the diverse population that is in this country and elsewhere. So there may be factors that are genetic, that are different, and how the body expresses or reacts to the pathology of Alzheimer's disease and other illnesses of the brain and neurodegeneration and aging in Latinos, for example, or in African Americans or in Asians. So I'll talk a little bit about that as well. We've dedicated a specific core to the Latino population because we're here in San Diego. We can really leverage our proximity to the border and the diversity of San Diego and really enhance the nation's research, really. I mean, even Mayo Clinic takes, they don't have a lot of diversity in Rochester, Minnesota. So we can help them make broader impacts because we send them our data. We send them, in fact, our human cell model tissue. They can make use of Latino or African American or Asian American cells and make progress that may be relevant or at least addresses potential differences that might impact how a person progresses with that illness, and also the social factors of that as well. Really important to address. And so we've made a real effort at our center to make sure that all of our research is broadly applicable. That's what we want to try to make sure we do. And then training the next generation of scientists. That's our research education component. So not only scientists, but also the workforce, the clinical workforce, the staff workforce. We need to make sure that our young, brilliant students in San Diego and elsewhere are ready to take on all the new factors that are taking place in this disease. And there's a lot to catch up on. I mean, because the field is moving very, very fast. So we need to make sure that we convey what we know to the next generation so that they can carry it forward and continue moving this forward. So I never really showed this slide in prior appreciation events, but I think I wanted to show you the breadth of the faculty involvement in making use of this data. These are all incredible faculty at UC San Diego that are leveraging what you give to us, the data and the participation. So we are at a phase where we are about ready to submit a renewal. So it's a great time to kind of step back and look at what we've accomplished over the past 5 years in this very large grant that comes from the National Institutes of Aging. It's about was about a $15 million grant. Now it's about a $23 million grant. So there's a lot of competition for this. We're gonna, but we have a really strong standing. I think everybody that's looked at our center has said, "Wow, you guys are enviable in what you do." But it still means that we're going to have to really buckle down and try to stay as state of the art as possible in pushing forward the science. So what are we going to be doing? Well, here's a couple of points. Not all of it, but a few points. We're really going to be leveraging biomarkers in this next phase. And as you know, and we've talked about it, not only our cerebrospinal fluid markers and our plasma markers, which you always have been very good about giving, we're going to probably step up the request for imaging markers. And those are the amyloid and tau imaging markers in the brain, where we scan you with a PET scanner and look within the brain to see whether those proteins of Alzheimer's disease are depositing, and because they deposit quite early in the process of this illness. So in fact, the National Institutes on Aging has initiated a harmonization approach, meaning trying to get all the centers to collect imaging in the same way so that we can really get a lot of data about this process of the disease. And also having that gold standard of imaging of the brain, it allows us to then use easier ways to get the information, like blood plasma, and allows us to compare it to the imaging results and say, well, what's matching that much more expensive test and much not that invasive, but just more challenging to get, because a PET scan is probably about $5,000 each time we do it. It doesn't cost you anything, but we pay about $5,000 every time we get one of those scans. And we're probably going to be asking for 2 of those, one amyloid and one tau scan. And we're going to do that on at least 24, probably more like 50 of our participants a year. So it's going to be, maybe all of you will get it. And I'll say that there's always been a lot of discussion about when are you going to return those results to us? And that's a fraught question, because these are research tools. And we've always kind of, when there really wasn't a clinical need to get these tests, we were really kind of cautious about returning information that wouldn't really change what you would do clinically. Well, the field's changing, as you're going to hear from Howard Feldman, and there's a real push within the NIA to say, prepare your patient, prepare your participants, but it's very likely that we're going to be returning this information to you with really careful counseling about what it means and what it doesn't mean. Okay, but it's really kind of exciting to be able to get this information about yourself and others. So amyloid PET, tau PET, MRI measures of our volume and vascular disease, which we're doing not only within our standard cohort, but really reaching out to the diverse communities, because there's not been a lot of MRI scanning quantitatively of the difficult to reach populations, for example, in the south border area. CSF was continuing to ask. Plasma, there's a lot of excitement about what just a very simple sniff test can provide in terms of things like the Lewy body type dementia. So dementia with Lewy bodies or other Parkinson's type related things. So we may be we're talking about implementing a quick sniff test to see how your smell is doing as you age as well. So this is a complex path of biomarkers, but really what it's meant to say is that these biomarkers that we're collecting can change in the progress of this illness as much as 25 years before any clinical evidence of the disease is shown. That's why it's super exciting for us to be able to potentially measure these different markers across time and understand where we can place an individual on this curve to say, are they at risk, at an early risk? What can we do differently to try to potentially intervene at that very early stage? And what can we intervene at, and what impact does it mean if they're at the sort of right side of this this time curve of how this disease progresses over 25, 30 years? It's a really remarkable length of time that we're now learning from these markers. And I won't go through all of these, but really we're learning that CSF and plasma can get us information that may place an individual on where they are in this curve. Okay? At the very right side is where the, well, let's start at the very left. That's before anything starts to go awry, talking 25, 30 years before cognitive symptoms develop, we will start to see some changes in plasma and CSF. That's really brand new research that's being out there. And the imaging definitely shows that as well. The amyloid starts to deposit. That's where the plaques start to show, and then there's inflammation that takes place as well. And then at the end of the progress, you start to see these tangles, these tau tangles that start to deposit in the brain as well. And that's when you start to begin seeing cognitive impairment that's taking place. Why are we still continuing to collect CSF if plasma is so valuable now? Well, the CSF is really the fluid that's bathing that brain and spinal cord. It gives us a tremendous amount of opportunity to discover new markers outside of these standard amyloid and tau markers of Alzheimer's disease. So there's just a quick example, and again, Doug Galasco is right at the forefront of this, called RT-Quick of synuclein, meaning I want to take a little seed of this bad protein of Parkinson's disease and put it in the fluid, and using really advanced testing, see whether it starts to aggregate the other proteins that are in that patient's CSF or that volunteer's CSF. And it turns out that if that synuclein starts to seed the other proteins to aggregate, it puts it shows that that person is at higher risk of developing a Parkinson-related illness. It could be Parkinson's disease, or it could be dementia with Lewy bodies. And you'll see in this little graph, the main point being the two columns on the left side are Parkinson's disease and dementia with Lewy bodies, and you can see that the measure is very different on that left side than it is in all the other either normal or other diseases. Normal controls, healthy controls, ALS, multiple sclerosis, Alzheimer's disease, Pick's disease, corticobasal degeneration, or progressive supranuclear palsy. All those are the subtypes of dementias that we need to disentangle from all these other, the umbrella term of dementia captures a lot more than just Alzheimer's disease, as you know. So, what am I super excited about? I'm excited about the new talent that's coming in. So I told you a little bit about Jerome Mertens, who is really excited and very engaged in our center and very interested in the human cell model approach. So when we approach you about maybe a skin biopsy to help us in our research, it's this individual that's going to be leading that science. He's looking at not only at when he turns that skin cell into a neuron via a pathway that makes it first into a stem cell. What happens in that case is it makes that cell like a baby again. So in fact, it's like you rejuvenating the cell and then turning it into a neuron. So now you can say, okay, well, what was this person's pathways of molecular pathways when they were a young child? What was it looking like? And then he also has another technique, and this is where he really innovated. It's a direct transformation of that skin cell into a neuron, which retains the aging impact. So now he's got basically a comparison of your brain when you were a baby with your brain when you were older. And he can start looking at, well, what part of this is genetics and what part of this is aging. So very, very exciting science that he's going to bring to the table. Now, bringing right close to him is a scientist from UC San Francisco who joined us, who is an expert in technology that you may have heard of, CRISPR. It's basically a molecular scissors to kind of chop up the the gene in any way you want and insert any gene you'd like. And it's really a remarkably powerful tool for discovering, well, what are the genetic factors that are going to influence those molecular pathways. So Jen Page joining us and really going to synergize with Jerome Mertens in our human cell model core. We also, as I mentioned, diversifying the science. We have a Chula Vista satellite site that you all hopefully know about. Makes it much more convenient for recruiting and collecting data in the South Bay. We're planning a launch of an Asian cohort for Alzheimer's disease. We're keeping an eye on that. And we're doing critical research on genetics and disease heterogeneity. So in order to do that, first step is to understand how these populations respond to us when we ask them for their genetics and their biomarkers. There's a lot of prior damage that we need to repair in prior scientific experiments that have been conducted in diverse communities. And so we need to rebuild a lot of trust and kind of understand, how do you feel about us being able to collect this information, including human cell model research and understanding what we're going to do with that data. We clearly training the next generation of diverse scientists, clinicians, and staff. This is really the 100-year journey of Alzheimer's disease, starting at the very left where it was discovered back in the early 1900s, and the huge amount of progress that's being made in these last 5 years. In order to convey that to our young scientists, we're going to need efficient approaches because a lot has changed in the field. You can't use the same techniques that we used to do when we were training our scientists. Just today, as Howard's going to tell you, we may be telling our clinicians something completely different on how do you convey biomarker results to your patients. And so this is really, really exciting, and it's a challenge for us, but it's a super exciting challenge. All right, so thank you to all of you. And we really could not do it without you. You help us advance the fight against Alzheimer's and related dementia. You continually sacrifice your time, your comfort sometimes, and you make that extra effort to bring your loved one in for us to evaluate, even though sometimes that's a challenge to get out of the house and get everything ready. We appreciate it so fully, and we just want to thank you so much for that. We really couldn't do it without you. So please, I just want to tell you a couple of other opportunities that are out there. Very simple to participate. The Gut Microbiome Project is basically looking at the bacteria in your stool and how that might relate to your clinical status. So pretty simple. Collect a little poop and send it to us. So that's a study you can be involved in. I think we compensate for that too, right? It's not super pleasant, so here's a compensation you'll get for sending us your poop. That's pretty amazing. Come to UCSD and you can make money off your poop. It's incredible. So hopefully that's some appreciation. Send us poop, we'll send you money. We have the standardized centralized Alzheimer's disease and related disorders neuroimaging. That is us asking you, please participate in our PET scanning and MRI studies that are available, and we'll be talking to you a lot about it. Same as the diverse vascular contributions to cognitive impairment and dementia, with a focus on the atherosclerosis that builds up in the brain with aging, and we quantify that, and we'll have information on that. May even return that. We're looking at returning of information. That may be an actionable information, like, hey, you may not notice it, but you're having little tiny strokes in the brain. Actually, little tiny strokes. And so we may need to control your blood sugar, your blood pressure, your cholesterol, all that kind of stuff. So it will be important to return this information back to you. And then social determinants of health. Those are some questions. I know that it's an added burden to ask some of these questions that might be personal. You can always turn us down and say, I don't want to answer that question. But we really do want to know some of the factors of what access do you have to health systems and many other factors that might be somewhat uncomfortable. If they're too uncomfortable, we never force you to do anything. You can always say, I'm not into that. I'm not going to participate in that. But it is part of our battery now to kind of understand these factors. We're sitting here in La Jolla. There's a lot of other factors that may be important if you're not in the super, high socioeconomic status place of La Jolla. So we'd like to know about that. So tell your friends, keep in touch, stay involved with us, join research studies, and help us in our push towards greater understanding and new therapies. Thank you so much.